Assalamualaikum and a very good evening to everyone. Before I start with my presentation, I would like to introduce myself first. My name is Nur Ali Anisa binti Muhammad Zaini. My number metric is E238-2X0. So next, my research title is Biomediation of Microplastic in Sungai Tebrau, Johor. Next, I want to start with my introduction. Microplastic in Sungai Tebrau has increased due to urban waste, industrial discharge, and population growth. This study aimed to assess the potential of biomediation of using natural microorganisms to break down microplastic. Water samples were taken from a resident area and industry area to compare pollution levels. This research is important as it offers a low-cost, eco-friendly solution to reduce microplastic contamination and support environmental sustainability. This research, I have three objectives. Objective number one is identify the level of microplastic pollution at selected location along the Tebrau River. Number two is study natural microorganisms from the Tebrau River water that may be able to live on the surface. Third, evaluate the efficiency of selected microorganisms in breaking down microplastic. So next, I present about literature review. Plastic pollution has been a concern since the 1970s, but awareness of its environmental impact has grown over time, especially regarding microplastic. These are plastic particles smaller than 5 millimeters that can come from cosmetic, clothing, and industrial waste. They are harmful because they can carry toxic pollution, harm marine life, and enter the food chain. These are some example of types of microplastic that pollute the sea that I found. Next, studies in Malaysia, like those on the Klang River, have found high level of microplastic, especially near urban and industrial area. Similar findings have been reported in international rivers such as the Yangtze from China and the Rhine from Europe, showing that rivers play a major role in transporting microplastic to the sea. Microplastic also provides surface for microorganisms to attach the form a plastifer, a biofilm of bacteria, fungi, or algae. Some species like Pseudomonas, Bacillus, and Aspergillus niger have shown potential to degrade plastic naturally. Biodegradation occurs in stage start from surface damage, biodeterioration to polymer breakdown, biofragmentation, and ending in mineralization, conversion to CO2 and water. Techniques like FTIR and Raman spectroscopy help identify the type of plastic and the effectiveness of degradation. Recently, studies have shown success using bacteria like Bacillus cereus and Pseudomonas protegens to break down polyethylene and polypropylene, showing up to 32% weight loss of plastic. Enzymes like PETase have also been engineered to degrade PET bottles efficiently. In conclusion, microplastic pollution in rivers like Sungai Tebrau is a serious issue. However, the presence of natural microorganisms that can break down plastic offer a promising, eco-friendly solution for biomediation and environmental restoration. Next, I will continue with methodology. I have two methodology like materials and method. So materials, I have two. One field materials and second laboratory materials. One, field materials. I have sterile plastic or glass bottle like 500 ml, rubber glove, waterproof marker, cooler box with ice pack, coarse mesh filter, notebook and pen, GPS device or smartphone app, microfilter and salt solution bottle and ACI solution. Two, laboratory materials like rubber gloves, aluminum flasks, micropipette and pipette tips, test tubes, agar plates, petri dishes, label and marker, forceps, filter paper, adhesive tape, storage jars, light microscope, glass beaker 250 ml and 500 ml, agar media, distilled water, saline solution NaCl, and sterile cotton swab. So next method. So I have a 7 method. So we start with one. So one site visit and selection. Two sites were selected using GPS from a resident area and one from industrial area for water sample collection. Second, river water sampling. River water sampling. River water sample will have been taken using clean 500 ml bottle at 0 until 30 cm deep. Glove will have been used and bottle labeled by date, time and location. GPS will have been used to record the site. Sample will have been kept in a cooler. A coarse filter will have been used first, followed by a fine filter to catch microplastic. salt water, and ACI will have been added to help microplastic float for easier collection. So, method number 3 is microplastic solution. Microplastic isolation will have involved filtering river surface water using a fine filter. A saturate NaCl solution will have been added to make microplastic fluid. The floating particle will have been collected using a paper filter and dry at the room temperature before analysis. Fourth method is microorganism isolation. Microorganism isolation will have been involved spreading 1 ml of river water on agar-neutral medium. The incubating it at 30 until 37 Celsius for 1 to 3 days different colonies will have been identified and transferred to pure cultures. This method have aimed to find microorganisms with the potential to degrade plastic. Method number 5 is microplastic biomediation test. The biomediation test will have mixed microorganisms with sterile microplastic and shaken for 7 until 14 days at 30 Celsius. After that, microplastic will have been filtered, dry, weighed, and analyzed using light microscopy and SEM to observe surface change. And method 6 is the surface morphology analysis SEM. The SEM analysis will have been used to observe surface change of microplastic. Dry microplastic will have been coated with gold, then placed in the SEM machine to capture high magnification image for degradation assessment. And lastly, for method is data analysis and reporting. Data analysis will have compared microplastic level at each site and measure how effectively each microorganism degraded the particle. So next, I will continue with flowchart. So flowchart start with site selection. I choose Sungai Tebrau, Johor. Next, water sampling. Next, sample preservation. I choose salt solution and ACI. Next, laboratory analysis have a two, microplastic detection and micro-solution. Next, biomediation testing. Next, using SEM analysis, I choose surface morphology analysis. Next, data analysis. And final is conclusion and recommendation. So next, I continue with Gantt chart and milestone. Gantt chart. In March, problem identification, objective setting, and literature review had been complete. In early April, site visit and sample collection had taken place. From April to early May, lab work such as microplastic solution and micro-identification had been carried out. This had been followed by biodegradation testing, assay matching, and data analysis throughout May. And finally, the report had been writing, and this presentation had been prepared by June. So next, milestone. The research proposal had been submitted on 11 March 2025. Site selection and sampling plan had been finalized by 20 March. Field sampling had been conducted on 9 April and lab analysis had followed. The biomediation and SEM stage had been complete by 7 May. Final report writing had been done by 22 May. And the final presentation, which is the season, marks the 100% complete on 12 June 2025. Next, I continue with research budget. For research budget, transport 100 ringgit for site visit, field materials total 160 ringgit such as sterile bottles, cooler box, labels and filters and others. So continue with laboratory materials, total in 290 ringgit, including agar plate, pipettes, petri dishes and SEM prick tools. And final, for research budget, is printing and miscellaneous RM 65 ringgit, which included reporting printing and unexpected expenses. So total for all this research budget is 650 ringgit. That's all for this. Before that, this is reference. 50 reference support my research. I think that's all for me. Thank you.